So in this lab, what we're going to be doing is taking some populations and breeding them under different circumstances, and then deciding if evolution happened or not. So in order to do that, we're going to have to compare the actual population that your group has versus what we would expect to happen in Hardy-Weinberg world. So remember, in Hardy-Weinberg world, we assume that those 5 major things didn't happen and that evolution didn't happen. So what we're going to need to do is to actually compare the frequencies that we have in the population that you mated versus what we expect to happen. Again, that's basically our hypothesis. So you're going to count your population, and then you are going to be comparing it to our expected values. And then you're going to pull out an old friend we talked about a couple weeks ago in your chi-square values. So the chi-square value, remember, is a value that we can use to help decide if our numbers are good enough to support a hypothesis or reject a hypothesis. So if you are still struggling a little bit with your chi-square value, no fear, you can still go back to lab 10 and look over it. Right? I suggest you practice with that a little bit as well, especially if you're feeling a little rough in that area, because you are going to need to be able to do the chi-square for this lab and then understand how to analyze that. All right, so remember with the chi-square, we did all the math, and I'll give you the tables for this lab as well, so you'll be able to follow it step by step across. But you are still going to have to compare that to the known value, right? So you're going to have to look back at the table that's in lab 10 to determine if your chi-square is an acceptable value or if it is rejected. If it's accepted, remember you have to look at the critical chi-square value. If it is below that value, then you can accept your hypothesis. If it is above that value, then you have to reject it. So you're going to be supporting or rejecting your hypothesis. Now also note that in this lab you are going to be asked one more question. You are going to be asked, did evolution happen or not? So remember, Hardy-Weinberg is hypothesizing that evolution did not happen, right? So if the Hardy-Weinberg equilibrium is supported, then evolution did not happen. We saw no change in our allele frequency. If, though, we reject that hypothesis, it means there was a change in the allele frequency that was significant enough that we know evolution did happen. So make sure that you are answering your questions correctly, right? Your hypothesis, was it supported or rejected? And then your next follow-up question is, did evolution occur or not? So make sure that you're understanding that a supported Hardy-Weinberg hypothesis means that evolution didn't happen. If we reject this hypothesis, again, evolution did happen. So let's talk a little bit about what you're doing in this lab. So you're going to start with establishing your population. So procedure 12.1 is establishing a known population. This known population is given to you in the lab book as the one that we are going to be working with. So this population is going to be 25% that will be homozygous dominant. So we're going to have 100 individuals, 25 of them will be homozygous dominant. 50 will be heterozygotes, and then the other 25 will be homozygous recessive. So that is 100 individuals. So if you do that math from the last video to figure out how many alleles there are, I will tell you, there will be 100 dominant alleles in your population and 100 recessive alleles. So in the lab, you're going to be given beads. These beads are going to represent the alleles. So I always say the red ones will be dominant and the yellow ones will be recessive. So you're going to create a population that has 100 red dominant alleles and 100 yellow recessive alleles. So again, remember that Hardy-Weinberg hypothesizes that evolution isn't happening. So this is what we are going to expect our population to be every single time that we have an event, that we have mating, anything that happens, we should always come back to this 25, 50, 25 population, always. So if we tie that back to something we've talked about before, this gives us a genotype ratio of a 1 to 2 to 1. So the genotype ratio of 1 to 2 to 1 is a 25%, 50%, 25%. This will be your hypothesis for the entire lab, right? Your hypothesis will not change no matter what. because the Hardy-Weinberg hypothesis is that we will not see evolution, which means we should always see or always expect a 25% split of our three genotypes. So let's look a little bit at what you're going to do with these three experiments. So this is you're setting up the population, and then the next three experiments we're going to do three different things to kind of see if evolution will happen under these circumstances. So 12.2 is going to be random mating. So during this experiment, you are going to put all 200 beads representing 100 people separate in one beaker, one jar, or whatever you're using. And you are going to reach in and randomly grab 2 beads at a time. So remember, every single person has 2 alleles, so this will represent one individual. And you're going to count them. So somebody in your group better get really good at tallying. You're going to make little tally marks to count your homozygous dominant, which would have 2 dominant alleles, which again, I usually count as 2 reds. Then you're going to look at how many heterozygotes you have. So if you pull a red and a yellow bead, that would count as heterozygote. And then the homozygous recessive would be 2 recessive alleles, which would be the yellow beads, at least in my brain. So you're going to count these, and you're going to continue doing this 100 times. So every time you pull out 2 beads, you will count them, and then you will put the beads back into the beaker and you will stir it all up again. And you're going to do this again 100 times. So you're basically going to take your 100 individuals and you're going to mate them until you have 100 babies out of that population. So in your lab book, you're going to see a chart that looks like this. In the purple box, in your genotype number, this is where you're going to put your final tally. So you're going to have, let's say 30 big A's, 55 big A little A's, and then however many little A's that is, because I just made that up. So you're going to have your number here. This is what you will count. Now from there, you have to calculate the other columns in this table. You will have to determine the frequency, which remember is taking the number of the genotype divided by the total, which in this case is 100. And then you are going to calculate the allele number and the allele frequency, which we talked about in the last video. So you're going to do that on this table. Then you're going to look at the next table you will see is here. So the next table should look familiar, very, very familiar, which is the chi-square value. So you're going to take your total number you counted and you're going to put that in your observed value. So that observed value will be what you counted. Then you will calculate the expected based upon our starting population of 25%, 50%, 25%, and then calculate your chi-square. And then the critical chi-square that you will get down there in the very, very bottom corner will be what you compare back to that table in Lab 10. And as a reminder, again, if that chi-square value is less than the critical chi-square value in your table, we are supporting our hypothesis that evolution did not happen. If that number is higher, then our hypothesis is falsified, it is rejected, which means evolution did happen. Now in this population, since it's going to be random, we should not see a very gigantic change in our population for this one. The next two, on the other hand, we're going to change something, right? We're going to change one of those 5 things we talked about earlier. So in the first one, you're going to be looking at natural selection. Basically, one of your genotypes is not going to survive to mate. So you're going to start with your base population, all 200 beads, right, the reds and the yellows all back in one beaker, and you're going to pull out 2 beads at a time. Now this time you are not going to put them back into the beaker. You are going to keep them and you're going to put them into 2 other beakers that you will have at your table, one that says dead and one that says alive. So any individual that you have that you pull out of that beaker that is dominant will survive, which means if they are homozygous dominant or heterozygote, they will go into the live beaker. That means they have a dominant trait and they can survive to reproduce. But all of the recessive phenotypes cannot live to reproduce. They will die. They'll go into the dead bin. Now you do not have to count these. Do not count them. You are just going to pull them apart, basically. Now, mathematically, about 25% of your recessive phenotype should die. So you put them to the side. Again, you don't have to count them. And you're going to put all of your remaining alive beads back into the pool, and you're going to mate them. So you're going to randomly mate them, reaching in, grabbing out 2 at a time, and pull them out. You don't put them back. You're going to reach in, you're going to pull them out until all of the beads are gone. So again, mathematically, about 25% of the recessive phenotype died approximately the first time. So you should have about 75 of the individuals that total here. Again, it could be a little more, it could be a little less, but you should not have 100 here. If you get 100 as your total, you got to go back and do it again because you did not do it correctly. So you're going to mate these and see what happens. Again, you're going to do the same thing we did in the last one. You're going to put in your numbers, you're going to calculate your frequencies, and you are going to do that for, again, your allele frequencies and your genotype frequencies. And then you're going to have to figure out if evolution happened or not. So you're going to use your numbers to transfer that over to the chi-square table that you have. You're going to figure out your expected. Again, remember your expected frequency is 25%, 50%, 25%. Only this time it will not be of 100, right? It will be of that approximate number, whatever yours is. Again, it should be about 75. It won't always be 75, but it'll be 25% of 75, 50%, and 25% of 75. Remember to add these together to make sure these two numbers in your total actually match. Otherwise, you'll want to go back and look at your math again. Make sure you did it correctly. You'll then calculate your chi-square as normal, and you'll compare that to the table back in Lab 10. So if that number is higher than your critical chi-square, then evolution did happen and your hypothesis was falsified. So the last one you're going to do, you're going to take all the beads and put them back into your beaker. All 200 of them should go back in. And you are going to reach in and grab out 8 beads. Those 8 beads represent 4 individuals that were able to survive a genetic drift event. Again, this could be anything. As I said earlier, this could be, a tsunami that wipes out a village. It could be a forest fire, were the 2 examples that I used. And only 4 individuals are left to mates. So you're going to select out 8 beads and you are going to mate them together 40 times. So just reaching in and grabbing out 2 at a time for 40 replicates. So you're going to again count your homozygous dominants, heterozygotes, and homozygous recessives, put those into the table for procedure 12.4, and then again calculate out your allele and genotype frequencies. And then you're going to have to determine if evolution happened. So you are going to calculate the chi-square one more time, where you're going to take your observed value and put that in the chi-square. Again, you'll have a total of 40 here instead of 100, so you have to do 25% of 40 for your expected of homozygous dominant, 50% for your heterozygotes, and then again 25% for your homozygous recessives. Again, make sure these numbers match to actually be 40. Okay, make sure of that so that your chi-square calculation will be accurate. So again, once you calculate that chi-square value, you will look back at the table. If that number is higher than the critical chi-square, then your hypothesis is falsified. Again, meaning evolution did occur. If it is lower than that, then evolution didn't occur because you supported your hypothesis. So again, remember, math-wise, you've got a couple of other videos that you can go through and watch the step-by-steps of how to figure out your frequencies, how to double check your chi-square values again, how to use your chi-square table. So do use those resources if you are still feeling a little uneasy about the math.